Hi everyone, my today's topic is Powering Life, the dance of chemiosmotic coupling. So, my name is Palak, I'm from group 322, and today I'm going to explain this topic through my presentation. So, let's start. Welcome to the world of chemiosmotic coupling, where energy and life intertwine. This presentation will explore the dance of ions across membranes, powering cellular activities. Join us as we unveil the mysteries behind this vital process that sustains life. So the first question is, what is chemiosmosis? Chemiosmosis is the movement of ions across a selective permeable membrane driven by an electrochemical gradient. This process is crucial for generating ATP, the energy currency of cells, and is a key player in cellular respiration and photosynthesis. It works on the theory that chemiosmotic coupling is the idea that the proton concentration gradients across inner mitochondrial membrane, which is generated by electron transport chain, is ultimately what drives ATP production via ATP synthesis. Mitochondria, often called the powerhouse of the cell, are where chemiosmosis occurs during cellular respiration. They create a proton gradient across their inner membrane, which is essential for ATP production through oxidative phosphorylation. Chloroplast and photosynthesis. In plants, chloroplasts utilize chemiosmosis during photosynthesis. Light energy is converted into chemical energy by creating a proton gradient in the thylakoid membrane, leading to the synthesis of glucose and oxygen. The proton motive force is the driving force of chemiosmosis. It is generated by the difference in proton concentration and electric charge across a membrane, providing the energy needed for ATP synthesis to produce ATP. ATP synthesis, the energy factory. ATP synthesis is an enzyme that harnesses the energy from the proton gradient to convert ADP and inorganic phosphate into ATP. The remarkable machine rotates as protons flow through it and exemplifying the beauty of molecular machinery. Chemiosmosis in cellular respiration. During cellular respiration, chemiosmosis takes place in the mitochondria as electrons move through the electron transport chain. They pump protons into the intermembrane space, creating a gradient that drives ATP synthesis. In photosynthesis, light energy excites electrons, which then move through the photosynthesis in chloroplast. This movement helps establish a protein gradient used by ATP synthesis to generate ATP, vital for plant energy. Chemiosmosis is essential for life. It fuels metabolic process in all living organisms. Without it, cells would lack the energy needed for vital functions, leading to breakdown of life itself. Applications in biotechnology. Understanding chemiosmosis has led to innovation in biotechnology. Techniques such as bioengineering and synthetic biology utilize these principles to create sustainable energy solutions and improve agriculture productivity. Now let's talk about the uses of chemiosmosis in human body. In the realm of cellular energy, chemiosmosis play a vital role. This presentation explores the intricate dance of chemiosmotic coupling in human cells, highlighting its significance in ATP production and overall cellular function. Join us as we unveil the mysteries. So, as you know, what is chemiosmosis? It is the movement of ions across a selective permeable membrane driven by electrochemical gradient. This is crucial for ATP synthesis in mitochondria, where protons flow back into the matrix, powering ATP synthesis. As you know, mitochondria often referred to as powerhouse of the cell and center to chemiosmosis. Proton gradient explained. The proton gradient is established by the electron transport chain, which pumps protons from the mitochondrial matrix into the intermembrane space. This creates a potential energy difference that is harnessed to produce ATP as protons flow back through a ATP synthesis. ATP synthesis is a remarkable enzyme that synthesizes ATP from ADP and inorganic phosphate. Oxygen plays a critical role in chemiosmosis as the final electron acceptor in the electron transport chain. Its presence allows for the efficient production of ATP, emphasizing the interconnectedness of cellular respiration and oxygen availability for life. Chemiomyces in another organism. While this presentation focuses on human cells, chemiomyces is a universal process found in many organisms, including plants and bacteria. Understanding its diversity helps us appreciate the fundamental nature of energy production across life forms. Several factors can influence the efficiency of chemiomyces, including temperature, pH, and the availability of substrates. Understanding this influence is crucial for optimizing cellular energy production and have implications in health and disease. Chemiomyces and disease. Disruptions in chemiosmotic coupling can lead to serious health issues, including mitochondrial diseases. Understanding these disruptions can pave the way for potential therapies and interventions, highlighting the importance of maintaining cellular energy homeostasis. Future research directions. The study of chemiomyces is evolving with ongoing research aimed at uncovering its complexities. Future studies may focus on therapeutic applications and enhancing our understanding of energy production at the cellular level, potentially leading to breakthroughs in medicine. In summary, chemiosmotic coupling is vital for ATP production and cellular energy. We have explored its mechanism, significance, and applications for health. Understanding this process not only sheds light on cellular function but also opens doors for future discoveries. Thank you.